In part 5 of this video lecture series for chapter 3, we are finally making it to the inside of our cells, and we'll take a look at some of the contents that we find inside prokaryotic and eukaryotic cells. And if we look inside, remember we find that there's these 4 characteristic structures for all cells that are out there. We can find our plasma membrane that separates the inside of the cells from the outside. We have our cytoplasmic fluid. We've got our chromosome and our ribosomes. And so we'll talk about this a little bit. When you look at the cytoplasmic fluid in the cells, it's mostly water. There's going to be dissolved salt in there, but also this is where the building blocks for making new cells are found. And also, when you look at this structure, the ribosome, that's your protein factory inside of the cells. And when you look at the structure of a ribosome, it actually is composed of 2 subunits that can come apart relatively easily. And both of these subunits are made of an RNA or multiple RNA molecules and multiple proteins that will assemble to make the small subunit and then the large subunit. And when you put the 2 of them together, you get your complete ribosome that is able to synthesize proteins inside of the cells. And if you look at this, people see 30S and 50S. Well, it doesn't add up to 80S. And the reason for that is this was actually an experiment done by a Swedish guy named Svedberg. And he would grind up the cells. So imagine grinding up all this stuff and then take that extract from there and put it on top of a tube with a really thick, dense, sugary solution there. And then he would centrifuge it, which means think of spinning this tube. And when you would spin these tubes, you're experiencing 1 gravity right now. And he would spin these tubes at like 300,000 times the force of gravity. And that would cause these ribosome structures to start to sink through that sugary layer. And he came up with a sizing standard for this. So again, think like a ruler, and that's where he got this 70 Svedberg units for measuring the size of this ribosome that is found in all these prokaryotic cells that are out there. And a Svedberg unit isn't just a matter of the size there, it's also a matter of the shape of the structure of this thing as it's sinking through that dense sucrose layer there. So you can see that these prokaryotic cells have this characteristic ribosome that you're going to find in all of the prokaryotic cells that are out there. And it has this 70S size right there. When we look in eukaryotic cells, they actually have a 80S ribosome that is also made of 2 subunits. But because it's 80S, we know that there is a difference in terms of the size and the shape of the ribosome that you find in all these eukaryotic cells that are out there. In addition to the ribosomes, we can also find that prokaryotic cells have this area where their DNA is condensed into what's known as the nucleoid region here. And typically in this nucleoid region, you're going to find your chromosome that is attached to the cell membrane. Unfortunately, it's not shown well in this picture, but you're going to find this single circular chromosome attached to the membrane there. And when we look in the archaea cells that are out there, they have proteins that are kind of like proteins that we find in eukaryotic cells, and the DNA actually winds around them. So they're known as histone-like proteins. And one feature that we can find in a lot of prokaryotic cells, not all of them have this, but a lot of them have it, is a small circular DNA that is actually independent of the chromosome. And this plasmid DNA can carry extra genes, and because it's smaller, it's easily transferred between the cells that are out there. And this is something we really need to be aware of, because often these plasmid DNAs will give cells a genetic advantage, like an antibiotic resistance. And so that develops in a gene that's carried on the plasmid, and then this plasmid can get transferred to other cells that are out there, because prokaryotic cells can harbor like 1 or 2 copies. Sometimes you can even get up to 300 to 500 range for your plasmids out there. So a few of those plasmids could get transferred from this cell to other cells, and then they could pick up that antibiotic resistance. So if you look on the inside of the cytoplasm for a prokaryotic cell, it is relatively simple in terms of the different kinds of structures that you're going to find inside of here, where your eukaryotic cell has these organs or cellular organelles that are kind of like the organs in your body out there that are not found in these prokaryotic cells. One thing that we do find is there are certain kinds of structures that you can find inside of these things, but they never have a membrane surrounding them. And these are some pictures just showing different kinds of prokaryotic inclusion bodies. And a lot of people, when they look up inclusion bodies, they find a report for a eukaryotic cell, but an inclusion body is basically an area where you've got a whole bunch of materials packed together. And for eukaryotic cells, that's where you actually see viruses will form inclusion bodies. But in prokaryotic cells, the inclusions are typically going to be storage granules. And here you can actually see little lipid droplets that are forming inside the cytoplasm of a prokaryotic cell. You can also find things like long chains of sugars that make glycogen, which is how we actually store up our sugar in our muscles. You can also see some volutin granules over here, which are also known as metachromatic granules, and this is where the cell is actually storing up a whole bunch of phosphates that it can use later on. And phosphates tend to be limited in the environment, and a cell needs to have this phosphate. If you remember back in the chemistry stuff, nucleic acids are going to need it, phospholipids are going to need it, proteins also are going to often have phosphates added to them. So this is something that these bacteria cells want to store up and save for later. You can also see these nice little sulfur granules over here in the figure that's labeled C right there. And this is again something that the cells are going to store up because they're going to use that sulfur compound for energy production. And so you can almost think of it like the cell is storing up its batteries for use later on. Some of the interesting kinds of structures we can also find over here is a little gas vacuole. And this is basically like a bladder for the cell. And it's going to have proteins that are forming this little hollow cylinder that can store gas that's going to be used to regulate buoyancy. So this prokaryotic cell is actually like a little submarine, and it can fill up these little cylinders with gas, and that's going to allow the cell to float at different depths in the water that's out there. And probably one of the coolest kind of things that you can barely see in this picture right here is what's known as the magnetosome. And if you look, you can see this really exaggerated picture. It's little iron crystals that we find here inside of these cells. And these kinds of spiral versions of cells can actually do what's known as magnetotaxis. So they can actually move in response to a magnetic field out there. And people first isolated these kinds of cells by literally taking metal rods and sticking them into the sand and hooking up a battery. And these cells were actually attracted to that magnetic field coming off of, or they would swim in response to the magnetic field. So after a few days, you go out there and you pull this metal rod out of the sand and you scrape it off, and you've got a whole bunch of these bacteria with the magnetosomes that have oriented on that because they think it's like the North or South Pole out there. The one thing that isn't pictured here is another kind of structure that we can find inside of some photosynthetic bacteria cells called cyanobacteria, and that structure is the carboxysome, and that's where they're actually storing up an enzyme that's going to be used during the process of photosynthesis for fixing carbon dioxide. So kind of cool, different kinds of storage granules that we find inside of cells where they're packing all these materials together. But again, there's no membranes around these kind of things to separate them from the rest of the cell. This is almost like you stuffing a bunch of things into the corner of a room. And you've got your computer is the brains in the room. And then this is just a whole bunch of snacks that you've kept there for use later on. One other structure that we can find inside of prokaryotic cells, and not all cells are going to have this, but you're going to find this in certain kinds of gram-positive bacilli that are out there, and this is a structure known as an endospore. And your endospore is basically dehydrated DNA with protective coatings around it that allows the cell to survive through adverse environments. So say the water dries up in summer, or say you live in Merced and it's really hot, those cells could die off, and what they do is they basically go dormant. They shut off and form this endospore. And so you can see some of the steps right here. The cell is getting bigger and it replicates its DNA, and it starts to form a membrane around that DNA, and this gives us what's known as the forespore. So if you look at this, you can see this DNA is already starting to get protected with the membrane, and it's going to form a couple of layers of membrane around here. And that is really just part of the cell membrane that is invaginating around the chromosome there. To make this structure. And then they're going to form a protective cortex around there. And this is where the cells are going to use calcium and other kinds of chemicals to form this almost bony-like material around there. And as if that isn't enough, having membranes and then bone around it, they make another protein coating all the way around the cortex right there. And this is what is actually being stained when you look at the endospore stain. That malachite green is actually slipping into the keratin protein, which is the same kind of stuff that you're going to find making up your skin. Your epidermal cells on the outside fill up with keratin, and this also makes up your hairs on your body. So the same protein that we have on the surface is what bacteria cells are actually using to cover their endospores, and it helps to make them a little bit more resistant to water. And this endospore is inside of the cell, and when the cell is dying off here, it will release what's known as the exospore that can then float around in the environment. And once it runs into water, that's going to cause this thing to basically break down the exterior right here, and that's going to allow the cell to go from this state where it's dormant back over here to an actively growing cell. That is what's known as vegetative growth as they germinate. So it's literally like a seed that is producing a new tree out there. So really kind of some cool structures that we can find inside of these different kinds of prokaryotic cells. But when we switch gears and look inside the eukaryotic cell, we can see there is a lot more stuff, and we'll take a look at that in the next video.